Okay, when we're talking about cellular respiration, we're talking about the inverse of photosynthesis. And so, where we saw that in photosynthesis, we were able to take a sugar molecule and we can draw it as a six-sided sugar, or we could write it as C6H12O6, just as we did in photosynthesis. And instead of making it, instead of being a product, this is now a reactant, and we're going to add oxygen to it, an oxygen gas, and we're going to end up producing ATP, which is the key currency for the cell. We're going to make CO2 and water, right? So ultimately, our objective, the whole reason why we eat the sugar, the stuff that we like to eat, and the reason we breathe in oxygen gas is to make ATP. The byproducts, the things that are made and leave the cell, are water and CO2. Okay, so now keep that in mind as we're moving through this, that this sugar molecule, the C6H12O6, six-sided sugar molecule, this hexagon, add oxygen gas to it. That's why we breathe in, and we make ATP and CO2. And of course, the CO2 comes out of our lungs as we exhale, and we also produce water as a side product. So as we take a look at this reaction, one of the very first things that happens is that we take glucose out of the bloodstream. But if you remember correctly from our discussions concerning glucose, concerning osmosis and diffusion, you should hopefully remember that oxygen gas passes easily through the membrane and finds a dynamic equilibrium. So as you breathe in, oxygen gas diffuses in the bloodstream. It gets captured by the red blood cells, and at the capillary level, the oxygen is released into the tissues, and that oxygen diffuses easily into the cell and finds its dynamic equilibrium. So the oxygen is available inside the cell. Secondly, though, when we consider this molecule called sugar or glucose, it's a rather large molecule. It's polar and usually designated as a hexagon because of the shape that it takes on, the ring structure it forms in solution. And so this glucose needs to get in. It needs a receptor. So it's a receptor-mediated in endocytosis. So you have this receptor that fits, and it's obviously not the scale, but it fits the glucose molecule, and then it would just take in the glucose and place the glucose inside the cell. Now that doesn't happen without some effort, and one of the steps to bringing in the glucose into the cell is the molecule insulin, and you've learned about that. We've learned about diabetes and its insulin's opposite, glucagon. These two hormones stimulate the DNA in the cell to produce this receptor. The receptor is made specifically to take in glucose because glucose can't go into the cell without help, and this receptor is the help. Okay, so glucose gets in. Once glucose gets in, it's easy for glucose to come out again. So one of the very first steps that happens when glucose gets in is it's phosphorylated. We'll take a look at what that means in just a second. We add a phosphate to it. And by now, after looking at ATP, after looking at photosynthesis, you should know that the way we do that is by taking ATP and coupling it with this molecule to make sure that the molecule doesn't go back out again. So in any case, what we have to do to glucose is break it down. Break down glucose, we call that the breaking down of glucose, we call it glycolysis. Glyco meaning for glucose and lysis meaning breaking up. The general outline of the processes that we're going to be talking about are here. First, we're going to talk about glycolysis. That's breaking up of glucose. We're going to make a three carbon molecule that we call pyruvate. And then pyruvate's going to have several places it can go depending. We're going to produce a little bit of ATP. It's kind of a side note. It's important for various reasons, but this is a big deal here, though, producing electrons. This is an electron carrier, NAD. You remember from photosynthesis uses an electron carrier called NADP. This molecule here, NAD, is what's used in cellular respiration. So in any case, we have NAD as an electron carrier taking that electron in the form of NADH to different processes in the cell. So these are kind of players you need to know. You need to know the player 1 being ATP, obviously, and we've talked about that. 2, NAD. We've talked about NADP, but it's an electron carrier. So 1, ATP as a currency of the cell. 2, electron carriers. And there's going to be 2 electron carriers that we're going to be talking about, and actually 3 if you count in photosynthesis NADPH. But here we'll talk about NAD/NADH, and we'll talk about NAD. I guess I should put NAD plus, and then FAD H2. These are your 2 electron carriers in this process, and they're going to take it to the electron transport chain, just like we talked about in photosynthesis. So those are your carriers. Those are the players that involve the carrying electron. You know you have sugar. Sugar or glucose is a major molecule that we have to talk about. So let's go ahead and add that to the list here. Glucose. You know we're going to have oxygen, gas, and CO2, gas. And of course, very much like in the process of photosynthesis, we have water. So really the players that we're going to be talking about are these 6, but we're going to add a couple more, and we're going to add lactate. And that's pretty much it. Those are, well, I suppose we should add, and it's an important one that we just mentioned a moment ago, pyruvate. So if you learn these 8 molecules, and a few of which you've already learned, right? You've already learned about ATP. You know about glucose. You know oxygen, CO2. You know water. So really all you need to learn is this NAD+, FADH2, lactate, and pyruvate, and the roles they play in respiration, cellular respiration. You'll be ready for this exam. So those are the molecules that we're going to talk about, those 8 molecules. Know them as we move through this process. As you're doing your cellular respiration, I think you've already completed, hopefully you've read your chapter. You should know these players already and be familiar with them, and you should have already probably filled out most of these in the question set that you should have done by tomorrow. However, when we take, those are the molecules, but there's 4 major processes that we have to be familiar with. Of course, the first one, as we said, is glycolysis. The second is either fermentation, or it goes into the tricarboxylic acid cycle, or another word for it is the Krebs cycle. And we'll talk about that in a second as well. So there's step 3, right? This is step 3. It's it's the same cycle. It's just 2 different names. And then the electron transport chain is step 4. And we've already talked about the electron transport chain. It's pretty much the same electron transport chain you saw in photosynthesis. All right, that being said, let's first talk about what happens in the cytoplasm. So glycolysis happens as soon as the sugar enters the cell. It's the first step, and it happens no matter what. The very first thing that happens with glycolysis is that sugar gets phosphorylated. In other words, a phosphate's added to sugar. You see that here 2 ATPs are used up, and 2 of the intermediates. And you need to understand something. You can look it up if you'd like. And I know it's in the chapter reading. This is not one step. There's actually many steps here that gets you to where you need to go. We're not going to go into that. what you need to know for your exam is that 2 ATPs are used up here, and this is what we call the energy investment phase. So you invest energy in order to get energy out. Now, that being said, this phase, phase 1 of glycolysis, and remember when we talk about energy investment, you need to think about what we've been speaking about already for a while, which is this idea of activation energy, right? So often in these biochemical reactions, you're going to invest some energy to get a lot more energy out. So we invest energy here at this phase in order to get energy out further down the line. So this is a 6-carbon molecule. Glucose is 6 carbons. When you're done with the first phase, the energy investment phase, what you end up with is 2 p-gals. And here we're down to 2 3-carbon molecules. Once you get 2 3 now of course 3 plus 3 is 6, so no one should be surprised by that. So one of the very first things you do in the second phase, this is the energy output phase. In this phase, what we're going to do, what your cell does, is it takes and releases an electron. You see here, you had NAD+, you had an electron and a hydrogen to it, and you get NADH. So this is before, and this is after. Now, it's important to think that this is a reactant, and you need NAD pluses in order to make NADH. So in this whole process, you have to think of it like a conveyor belt, like a game of hot potatoes. If you run out of hot potatoes, you lose. If they back up, if you get too many hot potatoes, you lose. So you need these reactants coming in, and you need these products leaving. So you need this product to go somewhere, and this is going to go somewhere, and we'll talk about where it goes in just a second. And it's going to go do something, and then what's going to return is NAD+. And this has to keep happening. This has to keep cycling in order to keep everything flowing. Otherwise, everything backs up. So NAD+ turns into NADH. That's going to get you some energy. That's a form of energy. We'll see where that energy is going to come out when we get to the electron transport chain. In fact, that's where it's going. It's going to the electron transport chain and the ETC. And that's where you're going to get energy out. It's going to get you a lot of energy, and you'll see how and why in a minute. What gets you less energy, but somewhat important, is getting these 2 ATPs out. This becomes extra important in fermentation, but you do get 2 ATPs coming out, and these 2 ATPs are important. It's very little compared to what you're going to get later. Now up here you lost 2, so I'm going to put -2 up here, and down here you have a +2. Now +2 what? ATPs, and -2 what? ATPs. But you have 2 3-carbon molecules, so guess what? This happens twice. This goes to the ETC, electron transport chain, and gets you a lot of energy, a lot of ATP out. And this is a +2 over here. So you have a net of +4, you see. But of course, you invested 2 to begin with in the energy investment phase. So what you end up with is +2 ATPs from glycolysis. So glycolysis, which happens here in the cytoplasm, it's important that you know where it happens. Here in the cytoplasm, this all is happening. And what you're doing is you're producing NADH, which is going to the electron transport chain, or to fermentation, by the way. And you're producing ATP. The ATP, how many ATP are you producing? 2. But wait a minute, I see 4 are produced. Well, you're actually producing 4, but you did use 2 to begin with, so net, to be fair, you're only gaining 2 ATPs from the breakdown of glucose to pyruvic acid. And of course, that's the other thing you make. You make 2 pyruvates, or pyruvic acids. So, And each of these is 3 carbons. So these 3 carbon pyruvic acids are sitting there ready to go somewhere. And that's the next step. Where are they going to go and what are they going to do? As I said, if the obvious answer is going to be it's going to enter the electron transport chain and do its thing. I mean, it is an electron carrier, NADH. And the pyruvate itself will go into the tricarboxylic acid cycle. But that's only assuming there's oxygen present. Now let's think about it. When will there not be oxygen present? Well, those of you that run track know that you start to run out of oxygen about what? As soon as you, I would guess around mile 2, you start to feel the burn, or maybe if you're in really good shape, the 8-mile mark. But in any case, at some point, you're going to start to feel the lack of oxygen. The better shape you are, the further you go before you start feeling that lower oxygen concentration. So when oxygen concentration is low, and that's going to depend on how much oxygen you're using, how well are you able to breathe in and out, that's going to determine in which direction this pyruvate that we produced from breaking down sugar, pyruvate that got produced from breaking down sugar, glycolysis, is either going to go, if oxygen is present, it goes this way into the TCA cycle in the mitochondria. Now, pyruvate enters the mitochondria. Everything else, the glycolysis happens in the cytoplasm, fermentation is going to happen in the cytoplasm, but in the mitochondria, you're going to have pyruvate go and do TCA and electron transport chain. So for this section, let's talk about what the rest of what happens in the cytoplasm. So we just talked about all of this, all the glycolysis steps, and where we get to pyruvic acids or pyruvate molecules. Pyruvate's the salt. Pyruvic acid is the acid form of the molecule. Let's take a look at what happens if there's no oxygen present. Now here's, by the way, a summary of what happens in glucose when it enters the cell, right? As soon as this is a cytosol, right? So this is the liquid part of the cell in between the cell membrane and the mitochondria. First 2 ATPs in the energy investment phase. Then when the energy output phase, you're getting NADH. This is going to one of the 2 places, either fermentation or the Krebs cycle. And here you get 4 ADPs are turned to 4 ATPs, and we just looked at the breakdown here, right? And you end up with 2 3-carbon molecules, 2 pyruvate molecules. They're high energy. They're glowing red because they have a lot of energy put into them, right? We've gotten a little bit of energy out, 2 ATPs, but there's still a lot of energy left to squeeze out of these molecules. And let's take a look at what we're going to do to get them out. So what's going to happen when you're running out of oxygen? You can't do anything in the mitochondria because to get that done, to do all the things that happen in the mitochondria, you need oxygen. And without oxygen, that won't happen. But your cell has to be able to live without oxygen for a short time. And the way your cell does that is by doing this thing called fermentation. In humans and other mammals, we do what's called lactic acid fermentation. So lactic acid fermentation, the salt of lactic acid is lactate. In yeast and in bacteria, some bacteria in yeast, they do what's called alcohol fermentation. So they do the same thing that we do here, where we take NADH and we use it up to produce NAD plus. And they do the same thing, but instead of producing this molecule of lactate that leaves the cell and goes into the bloodstream, they produce alcohol. Now, what kinds of sugars would they use? Now, if you think about it, if you take grapes, crush them up, and you put them in a bottle with some yeast and let them sit for a few years, what you're going to make is wine. It's those yeast are going to produce alcohol inside that bottle. If you do the same thing with hops, you'll make beer. You do the same thing with rye, you'll make whiskey. You do the same thing with rice. You'll make sake. So these yeast, all these different fuels or sugar sources, the yeast will make alcohol in this fermentation, using this pretty much the same biochemical pathway. In your cells, in our cells, we don't make alcohol, we make lactic acid. Now, lactic acid ultimately starts to back up. If it builds up high enough, could cause problems metabolically, and so this can't keep going on for too long. Some people connect lactic acid buildup to muscle pain. Others say different sources say different things, but lactic acid buildup is not a good thing for your body, but it's produced because you need to keep glucose to keep breaking down glucose to make enough ATP to keep the body going. Remember, without ATP, no cellular work is done. If there's no cellular work being done, we call that death. So we need to keep this ATP going, enough ATP to keep the cell running. Even if it's running low, it needs to keep running. So we need to make some ATP. Now remember, we need to use some ATP to make ATP. So even though we use 2, we produce 4. So we make it 2 ATPs net. Notice the word net here. But without NAD+, this isn't going to work. We're not going to be able to produce ATP. Now, the ATP can be used in work and make ADP. So this ADP will be prevalent, will be readily available. But NAD+ won't be if we keep making NAD+ turn into NADH. So we have to make NADH turn back into NAD+ so that we can keep this process going. Again, just like I said in the beginning, it's like a game of hot potatoes. If you don't have enough NAD+, you can't get the process to go. So you see here, NAD+ is replenished, made fresh again by this process of fermentation, which is the only reason we do fermentation when there's no oxygen, is to get enough electron carriers to take the electrons away so that we can produce ATP. Yes, it's not as so we need to keep NAD+. We need to keep it replenished. We need to keep the stocks of NAD+ ready so that we can keep breaking down glucose, so we can keep producing enough ATP to keep going, at least for the short term. And so that's where fermentation comes into play. When there's no oxygen, in the absence of oxygen, we do fermentation. We produce lactate. The lactate's released into the bloodstream. So pyruvate's turned into lactate. And it's done so, while it does that and then produces NAD+. That NAD+ then can be used in glycolysis to make just a little bit of ATP. All right, so that's the purpose of fermentation. And now you know what alcohol fermentation is, and you know what lactic acid fermentation is. You need to know that for the test. So I want to be clear that you need to know what's produced. You need to know that alcohol fermentation happens in yeast and bacteria. And you have to know that lactic acid happens in most other eukaryotic cells, but let's say mammals, for instance, and certainly in humans. All right, so know that, and know that lactate is the product of pyruvate turning into lactate and making NAD+. Know the purpose of fermentation, which is to replenish NAD+, one of the electron carriers, so that you can have glycolysis happen, so that you can make enough ATP to keep going. You're regenerating NAD+. That's the whole purpose here. All right, that's the whole purpose. Lactate leaves the cell, as you see in this diagram. It leaves the cell. So try to remember the diagram, try to practice it, draw it out a couple times, know what fermentation is. Remember, the test is on Wednesday. All right. So now the next couple of slides, I'd like to talk about the processes that happen inside the cell. But before we do, I want to make sure that we review what we've learned thus far in the video. We know that when glucose comes in, the very first thing that happens is it gets phosphorylated and enters glycolysis. You know now that Glycolysis is the breakdown of sugar. We know the major components, major steps. You don't need to know all the little steps, but you need to know everything you see here. That there's two phases. The first phase is the energy investment phase. The next is the energy output phase. That when the first phase you put two ATPs in the second phase you get 4 ATP out, producing 2 net. Okay, and you're going to use NAD pluses in both of these productions of all 4 of these ATPs. You're going to use NAD pluses to make NADHs, and then those NADHs are going to carry the electrons somewhere. You know now that if there's no oxygen, the electrons are going to be carried over here to fermentation. Pyruvate's going to go to fermentation, and the electrons are going to be carried to fermentation. Here they'll produce lactate, and then NAD+, which goes back to glycolysis, helping produce more pyruvate, and certainly enough ATP to keep the cell going for a short time. Lactate's going to be put into the blood, and that's the danger, is you can have lactic acid build up in the blood, and that's no good for the blood chemistry and your body chemistry. But it's necessary because we need to keep this going. If this was not possible without oxygen, you'd immediately die. So given those two options, the cell chooses to go ahead and allow lactate to build up in the short term. Once you start breathing, once you start increasing your breathing, increasing the oxygen intake, whether that means breathing harder or that means you stop exercising and sit down and relax, whatever the case may be, as the oxygen concentration goes up and you start doing the electron transport chain and the Krebs cycle, you start to produce, convert that lactic acid back into pyruvate, and it enters the mitochondria, and you break it up into a lot of ATP, and the lactic acid level goes back down again. So in any case, that feedback mechanism, that's how all that works, and all this is happening in the cytoplasm, glycolysis and fermentation. Okay, so that's done. Now, what happens when you do have oxygen, you have pyruvate? What happens inside this organelle called the mitochondria? Let's take a look. Okay, so as we're looking into what is occurring, all the different processes that are occurring, we have to remember that in the first step that happens in the cytoplasm, in the cytosol, is glycolysis. Glycolysis, the whole purpose of it is to take glucose and make it into pyruvic acid and make some ATP, and some electrons in the form of NADH get carried away, and those go right into the electron transport chain over here inside the mitochondria. Now, that pyruvic acid can go one of two places, and we just talked about it. If there's no oxygen present, it'll go over here to fermentation, which is not shown. And in fermentation, you're going to replenish the NAD pluses so that you can keep glycolysis going in a cycle until you're able to get enough oxygen. Glycolysis does not require oxygen, so it's anaerobic. But these processes in the mitochondria require oxygen. And you'll see where oxygen is required once we get to the end of the electron transport chain. But you have two processes going on here. The Krebs cycle, also known as the TCA cycle, which is happening here in the mitochondria, and the electron transport chain, which is happening here. And we'll look and see what happens in the Krebs cycle and why it's important. You see, you get 3 places you're getting ATPs. The most important place, the largest producer of ATPs is the electron transport chain. By far, it is the most important step for making ATP when oxygen is present, because it produces the most amount of ATP. And we'll see how much is made at each step in a moment. But the point is that the electron transport chain is, if you want to think about making shoes, this glycolysis and Krebs cycle makes shoes one shoe at a time, maybe a few shoes. It's your old cobbler concept that you have in the back room, an old man or old woman sitting down with the necessary resources, making a pair of shoes at a time. Where the electron transport chain is a factory model, we're producing a couple hundred, where these two can produce a few at a time. This electron transport chain produces many ATPs at any given moment, because it's literally a factory model of producing ATP. We call it chemiosmosis, and you'll see why in a minute. You've already been introduced to electron transport chain. You know how quickly, or you can. I hope you have an idea of how fast it's possible to make ATP using the electron transport chain. Let's look at what the mitochondria looks like. In fact, it reminds me a lot of the chloroplasts, except the chloroplasts, remember, they have these stacks of thylakoids, which is still an inner membrane, just like it is with the mitochondria. These inner membranes, they're stacked in the chloroplast. In the mitochondria, they create this matrix, right? This kind of puzzle inside folded membranes inside the mitochondria. The chloroplasts also have the folded membranes, but they form those granum, right? These stacks of pancake-looking things, where here this looks like a maze. There's an inner membrane here, as you can see. There's an outer membrane. There's an intermembrane space, which is this space right here in between the inner and the outer membrane. There are these things called cristae, which are the inner folds. You have embedded in the membrane all kinds of proteins that are necessary for all the processes we'll be talking about. They have their own ribosomes, which again suggests that they'll not only have their bacterial-like ribosomes, their own DNA. They have their own DNA in the mitochondria as well. Again, looking back and thinking about endosymbiosis in preparation for your end of course exams. But when you take a look at this mitochondria, these two processes are the important processes when you're thinking about a mitochondria and what happens across these membranes. Remember, one of the key aspects to any membrane is it separates two spaces, allowing one side to get a higher concentration of molecules than the other, which would then allow for what? Well, I hope you're saying osmosis or diffusion. And so when you have the high and low side across a membrane, then you can use that potential difference, that chemical potential, to allow for energy to be produced. And so we've talked about that concept in the sense of a battery in separating two sides, one positive and one negative. A very similar thing is going to happen here inside the mitochondria. So looking at the Krebs cycle, which would be here, the Krebs cycle is happening here, also known as a TCA cycle, so tricarboxylic acid cycle. So let's take a look at what these cycles look like. This is what the cycle looks like. Oxaloacetate is the last step, right? 4-carbon acceptor molecule regenerated over and over again in this cycle. You don't need to really know that, but another word for this, there's a Krebs cycle, there's TCA cycles, there's also citric acid cycle. And that's because citrate is the first molecule produced. These 4 combined with 2 more make 6, and this is a citrate molecule, 6-carbon molecule named citrate. There's all kinds of other smaller molecules and steps involved, enzymes. Each one can be controlled allosterically, or just controlled by products and reactants, levels of products and levels of reactants. But there's all kinds of enzymes producing all kinds of different things. You don't need to know any of that. What I'd like you to know for your exam, and the level at which I think you'll be testing on the end of course exam, is that you understand that these molecules that came from pyruvate, right? These molecules, there's a step that's missing. Pyruvate gets turned into acetyl-CoA. And when you do that, when you take this 3-carbon molecule, and it's not drawn here, but this 3-carbon molecule, and you move it through an enzyme, and you attach a CoA to it, this is CoA, coenzyme A, right? And you take this you cleave off one of the carbons, and you mix it up, and away it goes in the form of CO2. So that's your first molecule you lose. The first molecule of CO2 is lost, is made right there, right as soon as it enters the mitochondria, before it even gets to the TCA, Krebs, TCA, or citric acid. Cycle. All right, so it can be known as any one of those. So it all depends on who's writing the question. but all three are the same thing. Krebs, TCA, and citric acid cycle are, in fact, the same cycle. Just three names for the same cycle. I know. I wish people would just choose one and stick with it, but they don't. And so we have to deal with the fact that there's three different names for it. So the very first thing that happens at pyruvate is this CO2 gets cleaved off, and that's what first exhale you do is to get rid of this CO2, all right? Because this goes into the bloodstream, and your blood then exhales it from your lungs as you breathe out. So now, again, this is originally the carbon in the CO2 came from sugar. So this is actually what you've eaten. You're breathing out what you've eaten. So the next two molecules enter the TCA cycle. They get attached to oxaloacetate. What is it that you need to know? I think you need to know this. This is a very, very neat form of the citric acid cycle. It's not, there's not a lot here. If you know, it looks a little confusing, but there really isn't a lot. You should know that oxaloacetate accepts the first two carbons from pyruvate in the form of acetyl-CoA. I think you should know it forms the citrate molecule. As it breaks down, the very first thing that happens is that you take one of the carbons, you cleave it off, and you make CO2, and you produce NADH. As soon as you take that that carbon off and make CO2, you take an electron away. That high energy electron is going to go to the electron transport chain. So this is a lot of energy is going to come out of here. Okay, CO2 is gone. Again, you breathe it out, and then again it happens, and you have NAD plus going to NADH, and you have another CO2 going out. So what is it you need to know? I would write down, and I would make sure I knew that the first you lose. There were three carbons coming in, right? There's the three carbons coming in. The very first carbon leaves as carbon dioxide before the citric acid cycle, TCA or Krebs cycle, and then the other two, they leave during the citric acid cycle, TCA cycle, or Krebs cycle. The other thing you should know is that NADH, so one, for every one pyruvate, and that's pyruvate, right? Goes in, you produce 3 CO2 molecules, right? 3 carbon dioxides go out. The second thing you need to think about is that you're producing 3 NADHs. One here, another here, and another here. This is very important because this is where you're going to get a lot of your energy, NADHs, from the electron transport chain. And then the third thing is you're producing 1 FADH2, which again, just 1 FADH2 here. This is the fourth step, very important. And you're producing the equivalent of 1 ATP, but it's not an ATP, it's a GTP. And as I talked to you before, you can have a triphosphate of any one of the nucleotides, and this just happens to be one of the places where we do produce a GTP instead of an ATP. But energy-wise, it's equivalent to ATP. This will be able to phosphorylate a chemical reaction. You're able to couple chemical reactions to this GTP like you would with any other. All right, so that's really the TCA cycle. This is all there really is to it. It's just taking that pyruvate molecule and breaking it down into all its components, getting all the energy we can out of sugar. Remember, there's 2 of these, 2 pyruvates for every 1 sugar. There's 2 pyruvates per glucose. Now that's a really important idea, all right? So there's going to be 2 pyruvates for every glucose. So this whole cycle turns around twice for every sugar. So you're going to produce 6 CO2s for every sugar, obviously, because there's 6 carbons in a glucose molecule, so you'd expect to see 6 CO2s. You're going to produce 6 NADHs in the TCA cycle. Don't forget the stuff you produced in glycolysis. You're going to produce 1 FADH2. You don't produce any of that in glycolysis, so that's fine. You're going to produce 1 FADH2 per turn, 2 turns per glucose, so 2 FADH2s per glucose. And finally, 1 GTP per turn, 2 turns per glucose, so 2 GTPs. So after the 2 turns, for every 1 glucose molecule, you're going to produce 6 CO2s, 6 NADHs in the TCA cycle, right? And that includes, by the way, this portion out here in the preparation. This is the prep part, right? Just before you enter the TCA cycle. So we are breaking up this before. So you are producing one of these 6. One of these 3 gets produced before you even enter the cycle. Don't forget that. 6 NADHs, 2 FADH2s, and 2 GTPs. So I'll tell you, if you know all the things I just talked about, you'll be able to answer any question that I'm going to ask you on the TCA cycle, and any question I really think they'll ask you on the end of course exam. To review, what are the major points? First, you lose your first carbon in the form of CO2 before you enter the TCA cycle, inside the mitochondria, but before you enter the TCA cycle. 2, the thing that picks up the remaining 2 carbons that are attached to CoA, coenzyme A, is oxaloacetate. When it picks up those 2, it becomes a 6-carbon molecule, and it releases the CoA, the coenzyme. And this 6-carbon molecule is called citrate. At various steps, do you need to know step by step? No, not for this class. But you do need to know that you're producing NADHs, CO2s at each step. Notice that, you're at the first 2 times you produce NADHs, you produce 2 CO2s. And that's all the carbons. All the carbons in sugar are taken care of. They're done. So that's 1, 2, and 3 twice, because there's 2 pyruvates. 3 times 2 is 6. So that's 6 carbons, and they're all gone, and they all came from sugar. Now you can produce 1 GTP, which is the equivalent to an ATP. You produce an FADH2, which is another electron carrier, goes to electron transport chain, very similar to NH2. And then here you're producing 1 more NADH. So 3 NADHs, 1 FADH2, 3 CO2s, 1 GTP during TCA cycle, 1 turn of the TCA cycle. You turn it twice for every 1 glucose, so you double all of this here for every glucose molecule. If you know that, you know everything you need to know about the Krebs cycle. All right, so that's it for the Krebs cycle. Let's take a look at the electron transport chain, which should go faster considering we already covered it in photosynthesis. Based on everything we just talked about, you should be able to answer all of these questions. Now for the electron transport chain, let's take a look at this. By the way, if you want to try to answer the previous questions, I would make sure that pages 1 through 3 are done thus far, because the rest of these pages, we're going to talk about the electron transport chain. I'm going to go through the electron transport chain now. However, you should already have answered all the questions up to now, because I've given you all the answers in this short video. Also, you'll find the answers in the book, or you can ask questions during class. But this is due, and it needs to be correct. So make sure that you understand it. And remember, your test is on Wednesday. Now, I like color photos, so hopefully this helps. This is the intermembrane space that I showed you earlier. You can see the intermembrane space is space between the outer membrane and the inner membrane. So there's a space in between there, and that's what's here in blue. In the pink, which is inside the cristae, right? So the cristae are this kind of folds created by the inner membrane. Inside that space is this pink area, and you have these proteins that are embedded in the membrane. And what happens is that as the electron carriers, NADH and FADH2, you'll notice that NADH brings an electron earlier in the chain, which means you're going to get more bang for your buck from an NADH than you will from an FADH2. And you'll see that in a minute. And then when these electrons, these high energy electrons, enter the complex, just like we saw in photosynthesis, they get transported from one carrier to the next. The quinones are the ones that transport them from one complex to another complex. But in any case, they get captured up here. And as they do, just like we saw in photosynthesis, hydrogen ions get moved across the membrane into the intermembrane space, creating a high concentration of hydrogen ions on this side and a low on this side. And the only way you keep this high and this low is if you have electrons that keep flowing from the TCA cycle, Krebs cycle, or citric acid cycle. Remember, those are three names for the same thing. Or from glycolysis. In any case, the electrons get transported here into this membrane space, allowing for hydrogen ions to go from low to high. Where are you getting the energy for these things to go from low to high? The electrons. They don't need energy to go from high to low. So what you're doing here is you're charging up your battery. It's very similar to charging up a battery. You're moving ionic ions across a membrane, creating a high concentration of positive ions on one side, effectively causing a plus on this side and a negative on the opposite side. So there's your battery. Now, are there any negatives? Do you see any negatives on this side? Not really, except maybe for these electrons. But what it is that you have anywhere you have a high positive, the other side in relative to this side is negative. Okay. So as we said before with the other electron transport chain, if you have an electron here at the very end, at the very end, then you need to have something take that away. Otherwise, all these carriers are going to be full, and even though you're going to have NADHs coming here from the previous two processes, nobody's going to be here to take those electrons off their hands. And so these NADHs will never turn into NAD pluses, and soon the cell will run out of NAD pluses and the whole system collapses. So we have to have something to take these electrons. Well, that thing that takes the electrons, that stuff that you breathe in all the time, oxygen. So as you breathe in, that oxygen takes that last electron, mixes up with some hydrogens, and makes water. And so your final electron acceptor is oxygen gas. And that's going to be one of the key things you need to know. Oxygen gas is the final electron acceptor in the electron transport chain in respiration. Now, if you remember, in photosynthesis, the final electron acceptor is NAD+. So you need to keep those two things straight, because I promise you I'll be asking that on the test. So as you're looking at the final electron acceptor here for the electron transport chain in respiration, you're making water. So water is going to leave the system. It's going to go into your blood and you'll urinate it out or sweat it out. But in any case, water's made. You make your own water every time you breathe in. The oxygen you're breathing in, you urinate out or you sweat out. Okay? Very interesting to me. So this whole process, this whole chain is just as it is in photosynthesis, is just to create this high and this low, this battery. But then we have to use that battery to do something. And what are we going to use it to do? We're going to make ATP. And we're going to make a lot of it. So as I said, embedded in the membrane, and somewhere around here, if the membrane were continuous, and it is in the cell, but this drawing ends here. If this molecule, this protein, this series of proteins, actually it's a number of proteins, a complex, would be embedded in the membrane here somewhere down the line. And this is a continuous membrane. So these small groupings of complexes would be all throughout the membrane of the cristae, right? So when we're looking at this membrane of the cristae, this is one set of probably millions of sets of these. They're constantly creating this hydrogen ion high over here and low over here. And then there's these probably millions of ATP synthetases all in the cristae as well for every mitochondria, right? And I'm saying millions, it could be thousands. I'm not sure what the number would be. But the process is not just up to 4 little complexes. There's many, many complexes embedded in the cristae of the mitochondria. If you forget what the cristae, rewind the video and look at the anatomy of the mitochondria. So here's the membrane space. Here's outside. Here's inside. This is a high concentration of hydrogen over here. The hydrogen enters and fills in one of these slots. And when it does so, it turns this whole molecule, this whole area turns. The hydrogen ions come out here. Now these hydrogen ions can either be pumped right back into the high, or could be used here by oxygen to form water. So they're coming into this space where there's not a lot of hydrogen. They're going from high to low, and they're doing this through diffusion. But this hydrogen ion can either end up being part of water, or it could just be pumped, or it could just be attracted back over here and moved by the high energy electron into this from low to high, right? And where's it getting that energy again? Because this is active transport. Where's it getting that energy? From electrons, right? So in any case, this whole thing turns, and I'll be sending you a video, or we'll be watching a video in class concerning this. This whole thing turns. And every time it turns, what happens is these enzymes take ADP and phosphate and make ATP. And since there's so many of them, you can see it's very much a machine. You'll see it in class or in the video I'll send you. As these things just turn and ratchet, almost like a Gatling gun, right? It moves round and round and round, and it just keeps making these ATPs over and over and over and over again. Sorry for the sound effects, but that's what I imagine. That helps me remember. Putting it into context here, what we're looking at is this is actually wrong. Let me get rid of this. So looking at everything in context, if you think about it, what we're talking about here is the intermembrane space. This is the outer membrane of the mitochondria. Here's the inner membrane of the cristae. The citric acid cycle is happening here over and over and over again. Remember, two CO2s are leaving, oxaloacetate makes citrate, acetyl-CoA with the two carbons left over from pyruvate come into the cycle. NADHs are coming out of here. FADHs, you see them here, they're entering the system. They're carrying the electrons from the citric acid cycle to the electron transport chain, and away they go. Now remember, NADHs are coming in here as well from glycolysis in the cytoplasm. But all these NADHs, all the FADH2s, they just carry these hydrogen ions out here, creating on this side a high and this side a low. And of course, we know from high to low, the hydrogen ions are going to move from high to low. And as they move through, just like we saw up here, as you go from high to low, the ATP synthase is just going to make a lot of these ATPs over and over and over and over and over again, very much like a machine, making a lot of ATP. And this is the whole thing in context. It seems complicated from above, but I think if you think about it, and as you go through and answer the questions in the handout, the weekend handout, I think you'll be able, and if you've done your reading, I think you'll be able to put this into context. You have to do some memorization. You have to know what's happening at the citric acid cycle. You have to know where. You have to know glycolysis is happening in the cytoplasm. Citric acid cycle is happening in the mitochondria. The pyruvate goes either to the mitochondria if there's an oxygen. If there's no oxygen, you have to remember it goes into fermentation in the cytoplasm. If there is oxygen, oxygen is the last electron acceptor. As it accepts that last electron, it combines with some of the hydrogen here inside the cristae, and it makes water. The citric acid cycle is making those electron carriers. Those electron carriers go and move throughout the electron transport chain, charging up the battery, creating this high concentration of hydrogen on this side, low on this side. ATP synthase then is able to move through the membrane. Now, what you saw earlier was a similar diagram, but that was for photosynthesis. So you see the electron transport chain and ATP synthase happens in both the mitochondria and in the chloroplast, because both systems are being used to make ATP. Don't forget that. All right. In the case of the chloroplast, it's making ATP so that ATP can be carried to the Calvin cycle to make sugar. In the case of cellular respiration, it's to take that sugar, it's making ATP to use that energy for the cell to do its work. So two basic differences between the two processes, but the mechanism is the same. All right. At this stage, you should be able to answer all these questions based on the video that I just presented to you in the chapter reading You should have already been able to do it, but if you haven't by now, at this point, you should be able to. Where is the CO2 produced? I think I emphasized that. Where's the oxygen used? Where's the CO2? Where is sugar used and what's produced? I would even somewhere right in the side here write down what's made after glucose is broken down. Where's water produced? Where are the 38 ATPs produced? Where are 38 ATPs produced? Where's the NADH produced? The number of them in glycolysis, the number of ATP produced in each step. Add them up. Find out. 38 in total. Where are they all being produced? Their math should work out. By now, you should be able to answer all of these processes, right? You should be able to look at fermentation and figure out what's going on. Either you should understand alcohol versus lactic acid. I mentioned all that during when we discussed fermentation. All right, so if you're able to answer all these questions in your handout over the weekend and you're able to process this video, I know it's a little longer than I wanted it to be, but I wanted it to be complete. And of course, you have till Wednesday because Wednesday's the test, but the earlier you do this, the better. Make sure you understand both photosynthesis, cellular respiration, all the parts of cellular respiration. What are the electron carriers? If you break it down into the players and the processes, and then you ask yourself what's being made at each step and how does one process feed into the other, I think you'll make your life a lot easier. Also, if you learn the electron transport chain, it should work for you for both photosynthesis and respiration. You should think about the idea of charging up that battery and using the concentration gradient and that molecule ATP synthase to produce ATP. And remembering throughout all these processes, we're leaving a lot of it out for you to think about it in AP biology, but the idea is you're coupling chemical reactions using the energy of one released by one to drive another and make high energy molecules that then can be used to produce other compounds down the road. All right. So hopefully that all made sense. Good luck. I'll see you in class on Monday. Tomorrow, actually.